What is a protecting group? You have heard this term when you first learned about DNA synthesis. The protecting groups prevent undesirable reactions. In the case of peptide synthesis, what we call the FMOC protecting group is used today as a protecting group. You can see the structure of FMOC amino acid here. FMOC group is shown in red in this slide. Using the FMOC protecting group with amino group of the second amino acid that prevents the multiple coupling reactions. So now this FMOC amino acid cannot be coupled with each other. FMOC group can be removed easily under weakly basic conditions. We usually use piperidine to remove the FMOC group. So now you can see the coupling of the first amino acid and second amino acid. So now problem has been solved, but we still have a problem. That's the side chain problem. If you want to couple alanine with alanine, there's no problem. This is the only reaction product. This is the only reaction product you get. So what about coupling of aspartic acid? By the way, what is the one-letter code for aspartic acid? That's D, if you remember. Single-letter code of aspartic acid was D. Then what is the glutamic acid then? That was E. The smaller, the earlier in the alphabet. The aspartic acid and glutamic acid are also umami amino acids. You can remember these as delicious amino acids, DE, delicious. So let me go back to the peptide synthesis here. Aspartic acid has a carboxyl group on its side chain. So this means that the peptide bonding could occur either with the side chain or with the main chain carboxyl group. The reaction with the side chain would produce the undesired product as shown right here. So you can get two different products, but this is a desired product, but this is undesired product. Then to avoid the undesired reaction, the carboxyl group on the side chain needs to be protected using the tertiary butyl protecting group, which permits the formation of only one desired product. You can see the tertiary butyl group right here. That's why you cannot see the reaction with the side chain and main chain. This is the only product you get. That's how you can prevent the side reaction. In the case of lysine, the BOC group can be used to avoid side products. This protecting group is called BOC. It's often used to protect the amino group. So by the way, what was the one-letter code for lysine again? That was K, if you remember, which is close to L. So now let's summarize the solid phase peptide synthesis. Firstly, the first amino acid is attached to a base or solid support. Right? Then the first amino acid has an amino group at the end. You can add FMOC protected second amino acid. Then you can get dipeptide protected with FMOC. You can remove FMOC by weak the base, like piperidine, then you'll get free amine. Now it's ready for the next coupling. You can add the third FMOC protected amino acid. Then you can eventually get 1, 2, 3 FMOC protected. Then you can remove the FMOC protected group by weak base, and you can continue. So then this process can be repeated to complete the synthesis of the target proteins. Then finally, sample is treated with acid to release the peptide from the solid support and to deprotect all the side chains. These processes are actually simple enough to automate. In the next video clip, I will show you an automated peptide synthesizer.